ஹலோ. மை டர்ன்.
கேக்குது.
நீங்க
எனக்கு கேக்குது.
நீங்க ஆஃப் பண்ணிடுங்க மிஸ். வெளிய வந்துருங்க நீங்க.
வெளியில வந்துடவா?
ஆமா வெளிய வந்துருங்க. பேசு.
Hello.
அதான் வெளியில வந்து
Welcome back. Welcome back, everyone. It's time to enter into our second session.
I would like to call Ms. Renita Bensi of 2nd MSc Botany to introduce our resource person, Dr. R. Murugan, Assistant Professor of Botany, Ayyanadar Janaki Ammal College, Autonomous, Sivagasi.
Dear distinguished guests, respected faculty members, and dear participants, good morning to you all. It gives me immense pleasure to welcome you all to this National Lecture Cum Training on Taxonomic Tale of Plant Kingdom, organized by the Department of Botany under Star College Scheme, sponsored by DPT Government of India, New Delhi. We are honored to have Dr. R. Murugan, Assistant Professor of Botany, Ayyanadar Janaki Ammal College, Autonomous, Sivagasi, with us today. Sir has completed his UG and PG degree in St. Xavier's College, Palayam, Kotai, and PhD in Madras Christian College, Chennai. He has completed 12 years of teaching experience and 21 years of research experience. His area of specialization includes bioprospecting, medicinal and aromatic plants of angiosperm taxonomy. He has published 46 papers, 5 book chapters, authored 2 books, and edited 2 books. He has received 4 awards entitled Young Scientist awarded by the Science and Engineering Research Board, Department of Science and Technology, Government of India, New Delhi. CSIR Senior Research Fellowship awarded by CSIR CIMAP Lucknow, Gold Medal in MSc as an Academically Excellent Botany Student of the Year 2002-2001. And he received INSA CSIR CCSTDS Center for Cooperation in S&T among Developing Societies, Travel Fellowship in July 2007 to visit Kunming, China for attending International Trading Workshop on R&D of Traditional Ethnic Medicine. This conference is a platform for the students, researchers, academicians, and industrial experts to come together and share their knowledge and insights on the latest developments. Dr. R. Murugan is a renowned expert in the field of botany, and we are so privileged to have such an eminent person in our midst today. On behalf of the Botany Department, once again, I extend a warm welcome to all of you, and I hope that this conference will be a fruitful and enriching experience for everyone. Thank you.
Thank you, Ms. Renita. I invite Dr. Jacinta Tamil Malar, Associate Professor of Botany, to honor our resource person, Dr. R. Murugan.
Thank you, sir. Thank you, ma'am. Over to you, sir.
Thank you very much for the nice introduction. Good morning, everyone.
Good morning.
Most respected principal and head of the Department of Botany, St. Mary's College, my fellow teachers, research scholars, and students and participants of this college and other institutions. First of all, I would like to thank the organizers and the faculty and students of this department, this college, for a nice hospitality and welcoming me for this campus for the first time. I am very, very happy to be here and seeing the organizations of the lecture workshop and everything. So very, very, systematically it is planned and organized. So I thank you all for this wonderful arrangements, everything. And I also thank the organizers for inviting me and giving me the opportunity to share my little knowledge on the taxonomy to the young students. Students, I don't know whether the organizers knowingly or unknowingly organize the chosen the dates for this training workshop, since most of the more than 95% of the participants are the girls. Why I'm telling this is that you know that the coming 11th is the International Day of Women and Girls in Science. Students, the participation of girls and women in science is very meager, right? So though there is an improvement in the participation of women and girls in many areas, but still there is a long gap in the gender equality. So this seminar and aptly, the topic also selected. There is the taxonomic tale of plant kingdom. And the thing is that the girls' participation in the science of taxonomy is again very meager. So you see in the entire country, among a few taxonomists themselves, they are endangered. And among these endangered species, again, the critically endangered species is the women taxonomist, right? I don't know why, because there are many issues in science of taxonomy, and it is starting from the teaching taxonomy, because for teaching taxonomy, the teachers knows the taxonomy is very less, and the interest among students to learn taxonomy also it is very meager, right? There are reasons, the students or the people are thinking that there is no opportunity. But I would like to say that don't see taxonomy as the job getting science. But this is the fundamental science. This is a basic science, and this is mother of all science. Taxonomy. Because without taxonomy, you cannot learn anything else, any other branch of science, right? There are many experiences. We got people are visiting us, coming to us for identification and authentication of plant samples. They are bringing some samples and telling that this is I'm working on so and so species, but these plant specimens what they brought is totally different, right? And they are coming at the end of their PhD or end of their MSc dissertation work. So ultimately, whatever work you did or the people are doing is going to be wrong, right? They are thinking that they are working on some species, but ultimately they are landing in something wrong. So if you know a little taxonomy, you yourself you can identify plant initially, and later on you can confirm with the expert taxonomist. So that's why I am telling that this is the fundamental subject or the foundation for all or mother of all science. Taxonomy is. And today here to the pointer. Slide mover.
The pointer is not working. Work ಆಗಲ್ಲ.
Okay, I have my thing and sorry. Right. So today I am not going to teach you taxonomy. I'm not, because in 1 hour time nobody can teach taxonomy, because this is a practical subject and you have to visit a field for learning taxonomy. But this is just a fundamental tips I'm going to give you, right? How to learn taxonomy and other things only, right? So we are all taxonomists. Do you agree with me? Are you a taxonomist? Yes, we are all taxonomists. Even by birth, we are taxonomists, right? Because since the birth, we classify organisms, we classify whatever we see, the objects, isn't it? So basically we are taxonomists, all human beings, each and every one is a taxonomist. But I don't know why you feel difficulty in learning taxonomy, right? So taxonomy is nothing but just observation. So we are observing the objects, whatever we see, with morphological characters. We distinguish them based on the similarities and the differences. We classify the objects, whatever we see, animals, plants, or any other objects also, even stationary or whatever we see, we classify, even electronic gadgets also, right? So all this is why we classify, why we describe, why we characterize the objects is just for effective communication, that's all. We name objects. There is nomenclature, nothing else, right? So without name, there is no object in the world. Whatever we see, we name, right? So basically, we're taxonomists. And learning taxonomists, I think there won't be any difficulty, right? So taxonomy is the science that deals with mainly the collection of plant samples or whatever it may be, and identification, naming, and the taxonomy. So the man who classifies, identifies, names, and describes the objects, whatever we see, right? So basically, a taxonomist, we are all. Another thing is that the language is, we are all speaking some language, right? So language is nothing but whatever we think there is the medium of communication, right? So language is the process of knowing through the use of words. And it follows that the apt use of words are very, very important for the effective communications, right? So for describing a plant also, we use some words, that is the botanical terms, right? So the scientific names says nothing is a symbol of, say, communication, right? So to which most information is attached, right? Instead of describing the whole morphology, just we say one or two words, we can describe things. We are all given name just for communication only, just for the effective communication, nothing else. So this is the processes in taxonomy. Taxonomy is to develop a descriptive language for a precise characterization of plants, whatever we see, and devise a simple scheme for identification without describing the plants. So we cannot go for the further, right? So the identification, and we classify organisms. So work out a system of classification to show the relationship based on the similarities or the differences. And we have a method of naming, that is a nomenclature. So to naming all organisms. So these systematic activities, so these are the basic for the development of the subject of taxonomy. So before now going into the thing, the first thing is learning the plant morphology, that is phytography. So morphology is nothing but a tool for the plant identification, right? So here only the most of the students feel difficulty because the term whatever is used to describe the characters of the plants. So something you feel is a different language. So this is nothing the study of development, form, and the structure of plants. So we see the plants, we see the morphological characters of the plants, right? So based on that, we say this plant is this or the other one. So and by implication, an attempt to interpret this on basis of similarity and the differences and the origin. So this is the morphology. And plant morphology is the study of exomorphic characters of the plant, vegetative and the reproductive structure of plants. And these are used for identification, classification, and description. So if you want to learn taxonomy, first you have to understand the morphological terms. So there is very, very basic in taxonomy, right? So the diagnostic keys are the botanical keys we say, right? So both on a qualitative as well as the quantitative characters are useful for identification of the characters of the plants, right? So both characters are very, very important. Whether it is the qualitative characters and quantitative characters. So these both are now used in the classification. Next slide, please. So how to describe a plant? So there is a systematic manner we have to now describe a plant, and the technical terms have been developed over the years, and these terms are universally accepted. So botanical terms help in precise understanding of meaning and avoiding lengthy descriptions. So this is the basic of developing the taxonomical terms. So how to describe a plant? So based on a systematic manner or a particular sequence, like now the plant habit. So whenever we see the plant, we have to see the plant habit, whether it's a tree or herbs or shrubs or climber, so whatever. And we have to see the underground parts also. Then stem characters, leaf characters, then inflorescence, then flower, parts of flowers and fruits and seeds, right? And in each and every parts, so we have to see different aspect also. Like if take the leaf structure. So based on the leaf apex, base, margin, right, or the venation. So with all these characters we use for identification of our plants. Next slide, please. So then classification. So we classify, we arrange objects, organisms, or anything else for a greater command of knowledge and a more efficient use of information and an easier acquisition of data. So classification is nothing but the placing of organisms or the objects into groups and categories for effective organization and communication. So for this only we classify the objects, whatever. And the plant classification is the ordering of organisms or the plants into a hierarchy of taxa in positional compartments according to phenological characters, that is morphological characters and phylogenetic relationships or the artificial the criteria, right? So plant identification, sorry, plant classification, I think you have studied, there are a number of plant classifications been so far proposed and developed. So these, the classifications have been grouped into three categories. One is artificial classification and natural and phylogenetic classifications. So artificial system is the early developed one, and it's based on the number of small characters, like one or two few characters. And the natural systems reflect the situations as it is evident. It is believed to exist in nature, and it places all information available at the time of the classification. And phylogenetic system classifies organisms according to their evolutionary sequence, the genetic relationships, and it enables one to determine the ancestors and derivatives of any taxon. So whatever classification we study, even you can also develop the classification, but these classifications are grouped into these categories: artificial, natural, as well as the phylogenetic system. Recently, there is another system have been in place. So that is the APG classification. I think you may be studying. There is angiosperm phylogeny group. So which is based on the genetic sequences of plants. So among these categories, yeah, the artificial systems, there are a number of botanists proposed to this artificial systems. I'm not going into these things, and Theophrastus, Salpino, and Bohins. So there are many more botanists proposed artificial systems. Next slide. And among these artificial systems, so again, a number of botanists proposed to these systems, and among these system. Next slide. Bentham-Hooker classification is very important because worldwide and many classifications, many floras and revisions are written based on this classification. So this Bentham-Hooker, which although appeared after the application of the Darwin's theory of evolution, it is a pre-evolutionary in concept. That's why now people throughout the world followed this system for more than 150 years, and slowly in many herbarium and other the recent floras, they are following APG system of classification nowadays. So, next slide. So why I am projecting the Bentham-Hooker classifications here is that whatever floras we see today in India, all floras are written based on the Bentham-Hooker classification. So if you want to identify a plant based on the morphological character, so we have to follow the Bentham-Hooker classification only. So far no floras or revisions written in India based on the APG classification. So that's why I'm now bringing the Bentham-Hooker classification here. And I think you know this classification, and if you understand the classification, it is the plant identification is very simple, right? So Bentham-Hooker classified plants into 3 categories initially. That is deciduous seed plants, and dicotyledonate, gymnospermae, and monocotyledonate. So these are the 3 divisions that they made. And under dicotyledonate, again the 3 series have been proposed. Were proposed. There is a polypetale, gammapetale, and monoclamidae. And there are some. These the classifications are these divisions are based on a few characters. For example, polypetale, you know the meaning, 3 petals. Gammapetale, fused petals. Monoclamidae, there is no separation of petals or the sepals. It is the periantha, right? So based on this, they classified the organisms, so the plants, seed plants. Now you tell me that is it difficult to identify a polypetale plant for you? No. It's gammapetale. So by seeing the flower, you can say this is a fused petals or 3 petals, right? And the next one is again in polypetale. You can see the there are the series are proposed, right? That is a thalamiflora, disciflora, and caliciflora. Again, these the series are based on some characters. Thalamiflora means the flower parts are say attached on the disc-like structure, right? Or the disciflora, it is immersed in disc-like structure, right? And the caliciflora, that is stamens attached to the calyx. So these characters, based on these characters only, it is classified. Now you tell me that. Is it difficult for you? Is it difficult for you? Then in another case also, the gammapetale also, there are 3 series, inferior, heteromere, and bicarpellate. Inferior means it is inferior ovary, right? And remaining 2 series are superior ovary. I think you know what is inferior ovary, what is superior ovary, right? You know, isn't it? Right. So then under inferior, there are some the orders. Just you can go through that for each and every hierarchical taxa. There are a few characters. Based on that, you can identify the plant, right? And Keturumere and bicarpellate also similarly, right? And monoclamidae, again the 8 series were proposed by Bentham-Hooker, and in monocot also there 7 series proposed, right? Next slide. So if you want to understand these the important characters of these taxa level, so I put in brackets what are the important characters of those the series or orders, right? So as I leave this slide with you, you can share, you can just go through that, right? Otherwise, it is available in floras or books or in internet also it is available. So just you can go through that. Why I am focusing or giving more importance to Bentham-Hooker classification is that without this classification, you cannot use floras now. So the plant is given to you for identification. So based on the flower morphology, right, you can divide the sample into 2 or 3 groups, whether it is a dicot or monocot. And if it is a dicot, again you can identify whether it is polypetale or gammapetale, monoclamidae. So this is the way how you can use the floras. I think you know what is floras. Yeah, I think you have been handling the flora, using floras for your practical sessions, right? So if you want to identify flora plant using the Indian flora, understanding Bentham and Hooker classification is essential, is must. Without understanding this, you cannot use the flora. Okay, so just to go through the the Bentham and Hooker classification. That's why I skip the slides. Okay, now, okay, there are a number of families in dicards. Even without seeing the floras or classification, everything, by seeing a plant, right, you can assume. I'm not telling that you can identify. Remember, you can assume this particular plant belongs to some family, right? Based on some characters. What are those characters? By seeing whether it is armed or unarmed, right? So most of the plant species belong to the these families like Aporaceae, Rhamnaceae, right, or Balanites, Sarcinaceae. They are with arms, spines, thorns, right? You can assume that okay, these no, this is the no, the sample or the plant specimen may belong to this species. If it is aromatic, leaf is aromatic or the flower is aromatic, you can assume that the plant may be Lamiaceae, Laraceae, right? Or some other families like the Rutaceae. Identifying Rutaceae very simple. I will tell you some examples with a single character. We can identify the family. If you know the family, then using flora, easily you can identify the genus and species. Right? Provided you should know, you should understand the morphological terms, technical terms, like what is ovid, what is chordate, what is a cyme, what is a resin. So if you understand the meaning for these terms, you can identify easily. Okay. And similarly, then if it is a parasitic plant, so these are parasitic groups or the families, and the epiphytes, Orchidaceae or the Iaceae, and the aquatic plant. If you see aquatic plant, the aquatic plant may belong to these families, right? So with these a few characters, we can confine your sample to these families. Okay. Next slide, please. Just I want to show some examples how to identify a particular species, sorry, family, right? Because in the identification process, identifying the family is very important. Once the family is known, so then further you can identify the genus and the species. So you are given a plant sample, for example, and you are seeing many characters starting from the habit to leaves and all those things. But Rubiaceae family can be identified using a single character. What you are seeing is the interpetiolar stipules. Leaves are opposite. All plants in Rubiaceae, the leaves are opposite. And between the leaf, opposite leaf, you can see stipules. Whether the stipule is leaf-like or needle-like or spiny, so whatever it may be. So you can see the interpetiolar stipule. I think this light can be strong. Right? And another important character of Rubiaceae is the inferior ovary. Right? So these two characters, with these two characters, so you can identify the family very easily. Next slide. And similarly, the Rutaceae, again, you can see, the just if you look at the leaf against the light sunlight, you can find the number of dotted oil glands. You can see here, right? So oil glands are very common character in all almost all Rubiaceae species plants, right? So with this, that you can confine your identity or family that the plant may belongs to the Rutaceae family, and in the fruits also you can find the oil glands, right? And the Hesperidium, the fruit type is Hesperidium. Next slide. Brassicaceae, again, it is very easy family for identification. I think you all know the cruciform. What is cruciform? Cruciform. cruciform, you don't know, cruciform is plus. Okay. So if you see the flower, petals are four, they are in opposite direction, looking like a cruciform, right? So that is the meaning of a cruciferase. The botanical name is the family name is cruciferae. Old name is cruciferae. Now it is brassicaceae. So whichever flower you are seeing, petals four, it is in opposite direction, right? So you can identify that this is an cruciferae, and with the fruit character, you can confirm the plant family name, right? Next slide. And convolves again with a simple look. You can say this is convolvulaceae. How you can say this is convolvulaceae? If you see the petals, between petals, you can see some thickening here. See, there is a thickening. This is called plaquette thickening, right? When you are seeing a plant flower, it is a chemopetalic. That means the petals are fused. And between the flowers, petals, you can see some thickening like this. So these are thickened area, right? You see this plaquette thickening in the chemopetalic flower. This is a convolvulaceae family. So you need not to go through starting from the whether it is a polypetalic, monopetalic, all those things. Just from one single character, you can identify the family, right? Next one. So again, composite is very simple to identify, right? So Asteraceae, it's all head inflorescence with many flowers, and just you can go through it, right? Tubular or pen-shaped flowers, and the fruit is also very interesting. And then specialized fruits like dry fruit, dry nut, and often with the hairs on the top, right? Next slide. Again, Malvaceae, this is a distinct family, right? So the stellate hairs are present in most of the plant, and the mucilaginous sap. If you touch the plant, it is like the bendy leaf or fruit, right? So mucilaginous nature, and flowers with the epicalyx, right? So what you are seeing, the extra leaf-like structure below the calyx. So this is interesting character in Malvaceae, but the epicalyx is not present in all genera. There are two genera, Apotulum and Seda. So in these two genera, epicalyx is absent. So otherwise, the other characters are present. Okay. Next, and the stamens are numerous and other things. Okay. Next. And again, the apocynaceae. This is also very easy to identify apocynaceae. Like the plant often, not often, almost all apocynaceae members are with the milky latex, and the important character is the stamens, right? So stamens are five, and the anthers are fused and sometimes fused with the style head and form gynostegium. So this kind of gynostegium is present in particular families only, Apocynaceae, Archidaceae, and one or two more, right? And the seeds are often with tufts of hairs at the end. So this is another important thing. And these two families are, as per the Bentham and Hooker classification, both are different, Asclepiadaceae and Apocynaceae, right? But in APG classification, the recent classification, Asclepiadaceae has been merged with the Apocynaceae. Okay, that's why I put a mark here. Okay, next one. So again, identifying Lamiaceae is very simple, often aromatic, right? I'm using this word very carefully. Often, not all, right? That's why mostly aromatic. So non-aromatic Lamiaceae also present, right? And the peculiar inflorescence, right? That is luster. These are found only in Lamiaceae. And bilate. Almost most of the plants are bicarpellate. I think bicarpellate. And the chemopetalic, you can see the series of bicarpellate, right? So you can see bilipped flowers. Okay. So under the style is gynobasic. Gynobasic means the style starts from the base of the gynoecium, and the fruits are unique and nutlets are hidden inside the calyx. So with these characters, you can identify the family Lamiaceae. Next slide. And similarly, Acanthaceae also. These are two close by families. As the Lamiaceae and Acanthaceae. This also bilipped. Lamiaceae also bilipped. Berberia also bilipped. They are all coming under bicarpellate. But there are some differences. In Lamiaceae, the fruit is nutlets, right? here it is, fruit is in a capsule, can they see? And they see, the arrangements of, the things, and the seeds with the fruit-shaped jocculator, right? Peduncles are present in the fruits, right? So next. Yes, progeny. Again, the inflorescence is a specialized inflorescence like, the monothecial or swarfioid or helicoid sign, right? So this you can identify progeny with the hirsute or hirsute hairs, most of the plants. And again, this is also gynobasic style because these are close by families. Okay, next. So these are some of the examples of how to identify the plants with simple characters. I'm not telling that in all families with one or two characters you can identify the family, but some families without flora, right, with the field knowledge or field information, you can identify the family. Once the plant family is identified, then identifying genus for the species will be very easy for you. Okay. So the next topic is the plant nomenclature, and most of the students feel difficulty in understanding the nomenclature. And understand that without a nomenclature, without herbarium specimen, there is no nomenclature. Understand that. That's why in the afternoon we put on a herbarium training. And you are all going to do your project work, or you are going for further studies. And remember that whatever research you are doing, that you can do molecular biology work, biotechnology work, biotechnology work, whatever it may be. But don't forget to prepare the voucher specimen, herbarium specimen for your studies. If you are working on a plant, herbarium voucher specimen is essential. Without mentioning the herbarium voucher specimens, you cannot publish your work. You cannot publish your results or work in journals. If you send your results to a journal, immediately they will ask for voucher specimen. So voucher specimen is in the afternoon. I will tell you that what is that. And nomenclature is without herbarium specimen there is no nomenclature. And imagination, I cannot give you a new name to a plant. I can imagine, okay, so in that place I saw a plant, it looks like this, like that. So I can propose a name. No, in science it is not possible. We need evidence in science. So herbarium specimen is the evidence in nomenclature, right? So remember that herbarium is very, very essential, right, in taxonomy. Okay. So that's why I would like to give much importance to nomenclature, because understanding nomenclature is most of the students feel difficulties. But I will make it very easy. The next slide. Nomenclature, it is naming plants, nothing else. Okay. So names are means of reference. Why we are all given name? How will you call me then? Right. So the morphological description, can you call me? No, it is typical tall person with white hairs. Can you cite me like that? Refer me up. No, you cannot call me like that, isn't it? That's why now we are given one or two names. Binomial sometimes, right? Sometimes trinomial, sometimes polynomial also. If you go to other parts of the world or northern part of India, you can see trinomial and polynomial. I think you know what is all those things. Polynomial, more than 3 words. Trinomial, 3 words. In taxonomy, trinomial is there, but we often say binomial, right? But for subspecies and varieties, trinomial names are there. I will tell you that. Okay. Otherwise, it is commonly binomial. Okay. So the names are ordinary things vary from language to language and place to place. Even the common name also. So with the local name, uncommon name, or with a particular language name, we cannot communicate with other person in the world, right? So that's why the scientific names have been developed, and the scientific names are universal. Why scientific names are developed? These are for the uniformity and consistency. The scientific names are usually binomial. Okay. So I told you one person named Bahin, who proposed a natural system of classification. And often in many textbooks, so it is written that binomial name, who proposed a binomial name? Linnaeus, right? But whose idea was that? The concept was developed by Bahin brothers, Gaspar Bahin and John Bahin, right? The concept was they developed the concept binomial naming of plants or organisms, but it was implemented. It was proposed by implemented by Linnaeus for the first time in 1753 in Species Plantarum, right? So that's why Linnaeus honored Bahin brothers. They are twins, right? Gaspar Bahin and John Bahin. You know a genus called Bahinia, Tamil Mandarai. I think you know you have seen the leaf. The leaf is single leaf, but it looks like two. It's looking like twins. Leaf, right? That's why now the Bahin brothers are rightly honored by Linnaeus by giving the Bahinia name. Bahinia, the genus is named after the twin brothers, right? So in taxonomy, in naming, there is a meaning for each and everything. If a plant name is given, there is a meaning for the name, right? The name, it may be describing the species character, right? A species name may be given named after a place. Asadadachta, India, we can say it's named after the location, locality, or named after person. So for each and every name there is a meaning. Okay. So if you understand that, you will not forget. You feel the taxonomy very easy. So no, the varieties, I just now told you that for varieties and forms, no, subspecies, the scientific names are trinomial. Don't think that it is only binomial. Trinomial is also accepted for varieties. Okay. So the names are given according to certain internationally accepted rules, right? Unless there is no rules, we cannot follow a single pattern, right? When we travel in the road, there are some rules and regulations. So based on that only we drive or we go, right? So similarly for naming also internationally, there are certain rules have been developed over the years. Okay, so but it was Carl Linnaeus who proposed some rules for generic names of plants for the first time in this Genera Plantarum. And over the years, the rules are almost established by International Botanical Congress. So this Congress held every 5 years, right? The Congress only changes rules, frame new rules, changes rules, modify rules, see everything. So these rules, regulations, everything is coming as ICN. I think you know what is ICN. ICBN earlier. Okay, next slide. So for the set of rules of nomenclature were drafted by A.B. DeCandle first time, and as a base paper for discussion in the first Botanical Congress, International Botanical Congress at Paris in 1867, and these rules published as International Code of Botanical Nomenclature, and they still I see in many syllabus, right? This ICPN name is used. But this is outdated. This is 10 years back. They changed the name as ICBN as ICN. Okay, but this is the correct one. The International Code of Nomenclature for Algae, Fungi, and Plants. ICN nowadays, right? 2011 they changed it. So this is nothing but a set of rules. ICBN or ICN is nothing but a set of rules that govern the scientific naming of organisms. Nothing else, right? That's a scientific name. Why these taxonomists are always changing the rules and other things, recommendations, everything, to bring the naming system uniformity and consistency. That's why there are a lot of issues in naming, right? If you want uniform name, uniform system, so there are some the revisions, modifications, everything coming for stabilizing the nomenclature systems or naming system. Okay, so IUCN is used in two basic activities. One is naming new taxa, and another one is determining the correct name for previously named taxa. If a name is previously given, if it is not as per the rule of nomenclature, so we can change the name as per the rule of nomenclature. Okay, so these two are the activities or functions of ICN, nothing else. Okay, okay, next slide. So the code aims at the provision of a stable method of naming taxonomic groups. That's all, and avoiding and rejecting the use of names that may cause error or confusion. So, I will tell you some examples, right? And anyway, tomorrow afternoon you are having a training session for the solving nomenclatural issues, right? My teacher will teach you, train you. And today I am going to tell you the basics of nomenclature. Okay. So the aim of rules of nomenclature is that one name can be applied to one plant, and one plant, one taxa can be given only one name. So that is the main and basic aim of nomenclature. So there are some exceptions. You can say 9 families. So for example, cruciferae, just now we see that, and brassicas is the another name. If you give a name to a family taxa, the name to the rank of a family, there are some ends should be given, ACE, right? But these old names are not as per the nomenclatural rules. Cruciferae, there is no CEAE. But if you want to give if you propose a new family, so the ending must be CEAE. But these old names is not following the rule of nomenclature. But these names are also accepted as per the ICN, right? Because these names were in use even before Linnaeus, right? So since it has been these have been used for long years, so these names have been accepted, right? So the code has certain principles, rules, and recommendations. So we are going to see the principles and some rules. Okay, so next slide. So, and the principles of nomenclature, I think you would have studied in your classroom. So there are 6 principles, and this is the basic one. Nomenclature of the plants. ICN is independent of zoological and prokaryotic nomenclature. Right? The zoological nomenclature is different, botanical nomenclature is different. That is our first principle. And this is again important. The application of names of taxonomic groups is determined by means of nomenclatural types. Right? In taxonomy, when you hear this word type, it denotes the herbarium specimen, the original herbarium specimen based on which the name was given. Right? In the next slide, I will explain you what is nomenclature type. The next principle is a rule of priority of publications. Right? Priority of publication. Okay, there are 2 botanists, and for a single species, 2 different botanists propose 2 different names. Which one is accepted name? The confusion comes or starts. Okay, so there may be some dispute. My name starts in A, so you have to accept my name. A botanist cannot say like that. Or my country is a developed country, so you should accept my name. No, you cannot say like that. But there are some rules. There is no rule of priority. Who published first or published the name first? That name is accepted name as per the rule ICN, right? So that is a rule of publications, right? Or priority of publication. And the next one is already we see that is the basic aim of the ICN. That is at any position the taxonomic group should have can bear only one correct name. One species, one correct name. That's all. If any other names are given, that is a synonym, right? That is not accepted name, not correct name, but that may be used. Okay. Then the scientific names of taxonomic groups are treated as Latin. So problem for us is knowing the Latinized words, right? Almost all the taxonomical names are Latinized. That's why we feel difficulty, because this is in some other language. We don't know the meaning for many things. But you can get the meaning in various glossaries and other textbooks. So the final one is the rules of nomenclature are retroactive unless expressly limited. The meaning is that the rules apply to work done before the acceptance of these rules. This not only the rules are applicable only after the publication of rules, but it is even before that also in some cases. Okay, so these are the principles. Based on these principles, the other rules and recommendations are framed. Next is the priority of publication. So, okay, nomenclature types. So this is very, very important in nomenclature. Already I told you that on imagination I cannot propose a name for a plant. What is essentially is the herbarium specimen. So nomenclature type is nothing but this is the original herbarium specimen based on which the description of the species is written and the name was given. Okay, so names of families and lower taxa must be based on nomenclature types. What is nomenclature types? It is nothing but the constituent element, that is the herbarium specimen. So of a taxon or to which the name of a taxon is permanently assigned, whether as a correct name or as synonym. Right? The name was given based on the herbarium specimen. Then taxonomist going to the field and collecting plant samples, and after the descriptions, after identification, he comes to know that this is a new plant, new to science, new to the world. Nobody has so far described, so far published, not so far given name. So in that case, he proposes a name, right? So he describes, he gives the description, he draws everything, and if he has many specimens, based on a single specimen, so he has to give the name. That is called the nomenclature type. Next, I come to the types of kinds of types, different types, right? But this is the original type specimen based on which we give the name. Okay, so the nomenclature types has kind of very, very important role in taxonomy. For example, this example is given here for a family, and see, this is very, very important. Whichever words or the sentence I highlighted, these are very important. The names of families and lower taxa must be based on nomenclature type, not above the rank of family. For order, it is not applicable, right? Family, genera, and species, even below that, varieties, forms. For these, the nomenclature type is important. That means if you want to propose a family name, genus name, or species name, nomenclature type is essential. Okay. If you have any doubts, you can ask me even in between also, no problem. For a family, the genus, which is a type genus, again, the explanation is given below. For a genus, the type species. For a species, it is the type specimen. That is nomenclature type. Right? How your plan family name is given. So based on the holotype. Holotype is the original herbarium specimen based on which the description was made and the plant name was proposed. Okay, so this is for Lamiaceae, the type genus is Lamium, and for Lamium genus, type species is the Purpureum. The Purpureum species, the type is the holotype, that is herbarium specimen. I think you understand this. If you have any doubt, and even during the 3 hours also, you can discuss with me. Okay, next slide, please. So typification, what is a typification? There's nothing but type is a particular specimen, or in some cases a group of specimens. If it is a very minute plant, right? A group of plants are collected for preparing herbarium or specimens or anything else. Of an organism to which the scientific name of that organism is formally attached. So scientific name of every taxon is based on a particular specimen, or in some cases specimens. That is a minute plants. It is a group of specimens. Okay. So what you are seeing is type specimens. You can see here it is 10 typos. Okay. So the original specimens based on which, so this is very, very important. Understand this. This original specimen, right, based on which the name of species is given or designated as type. Even in this picture, you can see this is the original specimen. So this specimen was prepared, I think, 1830s. Okay. So based on this, the name was given Origanum platanoidus. The initial name was Origanum platanoidus, but later it was changed. I tell you the way it is changed. But this is the original specimen. Based on that, the plant name was given initially. That's why it is given the label as typos. Then types are usually a physical specimen. It may be a physical specimen, or in older time, illustrations were used for giving plant names, right? Based on the illustrations, based on the pictures, right? Or images, the plant names were given initially, but now it is a physical specimen is very, very essential. Okay, so since 1st January 1958, so before that, for valid publication of name at the rank of genus or species or below that, type is not necessary. You need not to mention the type name. But after that, after January 1958, if you want to publish a name, if you want to publish a species, so the physical type specimen is very, very essential. We should indicate. That's why I'm telling you that when the taxonomist is going to the field, they may collect 4 or 5 specimens. Among these 5 specimens, one specimen is acting as the original type, that is the holotype, nomenclature type. Remaining 3 or 4, that is another type. I will tell you what is that, isotype. Okay, so and another thing is, since 1st January 1990, for publication of there is valid publication is very, very important, of a name at the rank of a genus or species or below that, a single specimen must be designated as holotype. That's why I told you that maybe the taxonomist may have 4 or 5 specimens or more than that, and among these, based on one, he describes the species, or he designates a single specimen as holotype, that is the original type. Based on that, the nomenclature was given. Okay, next slide, please. Now there are now different kinds of types. I think you would have studied all those things in your taxonomy paper. Holotype, isotypes. I will explain to you one by one. Next slide. So what is the holotype? So this is a species. So one species newly described, not new. It is nearly 10 years old species of Pogostemon rahavendrini. Right. This is the specimen. The species was described by me during my PhD time. So I collected 4 herbarium specimens. Okay, collected 4 specimens, and later on critically identification, I came to know that this is a new species in Pogostemon. Right, the group on which I worked earlier. Even now also I have projects of Pogostemon. So among these species, as per the rule, I have to designate a specimen as a holotype. So this specimen I designated as a holotype. Before writing the holotype, I have taken this image. That's why there is no type name is given. So this specimen is a holotype, and remaining 3 act as the duplicate of the holotype. That means this one is original, based on which I gave the name, and these are part of the same collection, same locality, same date, same plant, everything same. So in that case, these specimens act as the isotype. Okay, so holotype of a name of a species is one specimen or illustrations either indicated by the author or authors in case of more than one, as the nomenclature type. This is very, very important. Nomenclature type is the original specimen. So used by the author when no type was indicated. So before that, no type was given, no name was given. So this is the first one. Next slide. And this is isotype. Isotype is nothing but I had 4 collections. So one was a holotype, and remaining 3 are duplicates of the original. That is called isotype or holotype. It is. So this is the duplicate of the holotype. Right. Next slide. So then paratype. Sometimes taxonomists not only collecting the plant samples, but at the time of describing a new species, they may study the collections made by other taxonomists. Right. So in such conditions, the founding author, that means the person who described the species, may have different collections, right? 2 collections, 3 collections. These collections may be collected from different locations. So based on these specimens, maybe 10 specimens or 15 specimens, he may come to the conclusion that the particular specimen is a new species. In that case, he has to give a name. But he has collections of different collectors, collected different date, different locality, everything's different. Then among these collections, he cannot say one is holotype, remaining all are isotype. Isotype means the specimens collected along with the holotype. But other specimens also used for describing species. So in the conditions, the other specimens are designated as paratype. If you have a doubt, you can ask me. Otherwise, that's how I explain. One example is given here, right? Jain and Hemadri. They examined numerous specimens of a particular grass. There is a person working on grass here, collected by Gami from Kasur Rock in Mysore, near Mysore, and by Talbot. This is another taxonomist. These two are 2 taxonomists, right? Collected specimens from 2 different places. One is from Castle Rock, one is from Amblighat in Mysore. 2 location, 2 collectors, 2 botanists. So they labeled Gami. Who labeled Gami collected 6 specimens. Dalport collected, say, how many Dalports here? Where is Dalports? Okay. This one, what the specimen is not given. But Gami collected 6 specimens. So Jain and Hemadri, while describing the species, they studied both collections, Gami's collections and Dalport's collections. They named Gami's collections as the A, B, C, D, E. That means 6 specimens. Among these 6 specimens, they proposed a name based on the Gami A. Then Gami A acts as the holotype. Remaining 5 specimens are the isotypes. So they designated the A as a holotype on their new species Manisurus mysorensis, that is the species name, and the remaining specimens of Gami, the 5 specimens act as become isotypes. But the Dalport collections also they studied, not only Gami's collections. So they, Jain and Hemadri studied the Dalport collections also, and based on that also they describe the name, a new species. But they cannot designate those collections as isotype. Yes, these are part of other collection, other collector, other locations also. So in that case, the Dalport specimens are not duplicate of holotype, but these were studied by the Jain and Hemadri, and therefore they are paratype. Right. So paratype is the specimens studied by the author, founding author. Founding author means the person who named the species. Right. Other specimen act as the paratype. Understand? Next slide. Syntype. Again, before 1958, designating a type is not essential as per the rule. So no holotype or any type is were not designated by those botanists, and specimens are studied by founding author and form the basis of new species or taxon. But the author did not designate any specimen as holotype. We don't know. Right? For example, this is a species Pogostemon nilogericus. So whatever examples I am showing here, all are most of them are Pogostemon genera, right? Because I worked on that. That's why I brought here. So this is Pogostemon nilogericus, and this was described by Gamble. Who is Gamble? I think you know, the person who wrote the Flora of Presidency of Madras. You are using your practical classes. So who Gamble described this species, but the collections were not by Gamble. Some other botanists collected from different locations. But Gamble did not designate type, holotype, isotype, paratype, nothing. But There are some collections, 4 or 5 collections. Based on that, he designated the species. He proposed the species. But later on, botanists, they come and they see that there is no type, no holotype, no isotype, no paratype. And nobody knows based on which specimen he gave that name. So in that case, all 4 or 5 specimens becomes syntype. Okay, so that's the meaning of syntype. So if the author studies collections from different localities and by different collectors, and decides to establish a new species, labels all of them as types, and all these specimens become syntype. Okay, so that's the thing. Next slide. The next one is lectotype. Okay, type was designated, but it was lost. What should I do? Then we can select a type, but we cannot mention as holotype or isotype, or syntype or a type. If a type is lost or destroyed in some conditions. So specimens are illustrations designated from the original materials as a nomenclature type if the name was published without a holotype or if the holotype is lost or destroyed. It is a type chosen to serve as a holotype. It is a type chosen as holotype, but it is not holotype, right? So it is written as the lectotype. Even in this, you can see lecto. This minutely you can see this type is lecto. That means the original specimen type specimen is lost. It's not available. So it is types chosen to serve as a holotype when either an earlier designated holotype was lost or destroyed. A specimen is chosen from isotype, paratype, and syntype. These are available, but holotype is not available, right? So to serve as the holotype, an expert, a taxonomist can choose a holotype. It is not a holotype, but it is lectotype. Understand that, right? From these collections. Next one. Then these are some of the other types. So when all these types are lost, holotype, isotype, paratype, syntype, all types lost in some occasions. In any other thing. So yeah, new type is selected from the specimens to serve as type. So this also a taxonomist or expert can do that. And the topotype is when no original type material is available or specimen collected from the type locality is chosen to serve as the type. So everything is lost. So during World War II, right? So many herbarium specimens were destroyed in European countries. In such conditions, I think they collected the samples again from the original locations, and they some specimens were designated as the topotype. So there is another type called epitype. So this is whenever the ambiguous or the confusions occurs. So that cannot be critically identified for purpose of the precise application of names. So these types are designated as the epitype. So these are the types of kinds of types. So based on that, the nomenclature is given. Next slide. So rule of priority. So each family and lower rank taxon with a particular circumscriptions, positions, and rank and barely only one correct name. That is one of the principles of the taxonomy. So special exceptions are made for 9 families. Just I told you that, right? Exceptions are there. But the priority of publications is that when 2 or more competing possibilities, right? That's why I told 2 botanists proposed names and 2 different names for a single species, one species. So in such conditions, the rule is applied. Okay. So for a name, the one published first is the correct one. The example is given here. So among these 3 names for a single species, Pogostomon henianus, this published by Bantam in 1830, Pogostomon suis in 1847, and Pogostomon among these 2. Which one is earlier is the 1830. Right, that there is no Pogostomon henianus that has the priority and is the correct name. And tomorrow you are having the training session. Please see all those things. Based on this only, the nomenclature will problems will be solved. right? And the principles of, okay, so which one is the starting point? There should be some starting point if we talk about the rule of priority of publications. On which date onwards, I can say this has a priority, right? So for that, the principle of priority for vascular plants starts 1st May 1753. So what is the date? The date on which Linnaeus published Species Plantarum, right? Linnaeus introduced or proposed the binomial system of classification. Sorry, the binomial system of nomenclature. Okay, so that's why this point has been taken as the starting point for angiosperms. For different groups, there are different dates, starting dates. Okay, for angiosperms, this is the starting date, and after this date, the rule is applicable. And before that, suppose a person published 30th April 1753. So just one day only. Give exceptions, no exceptions. So before that, whatever names published, that is not accepted name as per rule. Okay. So the names published prior to that are not considered for priority. Next slide. So priority of applications only applies to the taxa at the rank of family and below. Before that or above that, order, series, or not divisions, for that the rule of priority is not applicable. And for any taxon below the rank of genus, for correct the correct name is the combination of the final epithet of the earliest legitimate name at the same rank with the correct name of the genus to which it is assigned. When a name is changed, for example, I showed you a plant picture also, Origanum, the name is given here, Origanum bengalensis. This was the first specimen in Pogostemon. Okay, so Pogostemon, sorry, Origanum bengalensis, this Burman F, Burman filial, that means father and son. There are two botanists. So when transferred to the Pogostemon, so first the Burman F who described a new species and designated the name, given the name Origanum bengalensis. Bengalensis is named after the place. But later, when the Kunz studied in the Pogostemons, who came to know that the particular specimens studied by the Burman F is not Origanum genus. This is some other genus. It is different genus, Pogostemon, right? So he transferred this name to the Pogostemon. So in such conditions, when transferred, one plant is name is species is transferred from one genus to another genus, Origanum to Pogostemon, the specific epithet species name must be retained as per the rule. And the original author who named the first name should be within the bracket, Burman F within the bracket, and the this person is newly given the name, right? So this is how to solve basic nomenclature problems. Okay, so like that some are coming, and similarly this is one case. This is within the family, right? But here another case that is within the same rank species. From species only it is for changing genus to another genus. But another case is a name has no priority outside the rank. This is between the rank, and when it is changed outside the rank, but that is from the variety or subspecies to the higher rank. For example, Magnolia virginiana variety fetida, Linnaeus published. When this fetida variety raised to the species level, next botanist comes, that is the name is I don't know. Who studied and he realized that okay, this is not the variety, it has the eligibility to become a species. So in that case, he need not to maintain the epithet name. Fetida is not necessary to maintain. He can give a new name because it is from the variety to the next level. So within the same rank, the epithet name should be maintained, right? Otherwise, it is not necessary. That's why he gave the name So instead of not Fitida, right? He gave the name as grandiflora, Magnolia grandiflora, not Ripitida, because it is given the rank. So for purpose of priority, names of fossil taxa compete with the names based on the fossil type only. So these are not something. Okay, next slide. Okay, effective and valid publications. When new names are given, just another 5-10 minutes. To new taxa, when it is realized that an existing name of a plant is in contravention of the rules and it is illegitimate. And for new combinations, the combinations is given here. I am bringing all these now single examples in many places for your easy understanding, nothing else, right? So for describing a new species, we are giving new name, right? And when there is a confusion in the name, or it is as per the nomenclature, we give a name. Or when a new combination comes, new changes comes, modification comes, we give a name. At these now conditions, we propose new names. Next slide. So effective publications means that should be published effectively, right? So any printed matters when widely circulated is regarded as the effective publications. Okay, so if it is in a printed form, a PDF on January 2022 of electronic materials, PDF form in an online publications with the ISBN number or ISSN number. Without this number, if you publish any new species, it is not accepted. Next slide. Valid publication means when it is valid, name must have proper form for its rank. That means if it is a family, it must end CEAE for order ALS. Right? I think you know that. The name of the author and the rank must be given. That is species or genus or whatever it may be, and it must be accompanied with a full or at least a description in Latin. So it should be Latinized, at least a synopsis of that. The nomenclature type must be indicated, and the date of valid publication is the date on which the effectively published material is made available. So these are the valid publications. So if you publish a new species, you have to follow all these things. Okay, next. So author citation again. So author citation is very important in publishing names, right? So in all botanical names, after the binomial or trinomial, you can see some names, right? Linnaeus is L, right, or LN, right? So if it is a single author, you can find a single author name after publications. If it is next slide, if it is more than single author, two author or multiple author, so one author and you can use yet or and, right? That means two authors. If it is more than two authors, so all author names must be given, and between the two authors, last two authors and or yet should be used. And when we use parenthesis, when the name is changed, right? When we retain the species name, in that condition, the original author name must be retained, and the original author name is given within parenthesis. Okay, so that is the examples also is given here. Next slide. Okay, use of X. That means when an author had proposed a name, but was validly published by another author. This is very, very important. The name was proposed by someone else, but it was effectively published by another person. So in that case, so the X is given here. Gonika was proposed the name, but is not published, validly published. So in that case, Linnaeus validly published that name. That's why these two are between these two names X is given, and they use Lin. That means one author published the species in another author's say flora or any other manuals, right? This is Wallich's flora. He published niche published this species. And the use of a square bracket is square brackets are used to indicate a pre-stating point author. That means so Linnaeus published this name Lupinus, but the name was proposed or used by Torn40. Right, that's why the within a square bracket Linnaeus used this. That means the name was not Linnaeus, Tornfort's, but before starting point. He used that name, but it is after than the starting point he used that, published that. That's why in such conditions, but this kind of no, is very, very rare to see. Okay. So next slide. So retention of name, when it is retained, names are retained. Okay. So when I already told you that when the specific epithet is changed, when we transfer the species from one genus to another genus, the specific name is specific epithet is retained, right? So this retention of names, and if this amendment, listen, this is very, very important for now many genera or many families are merged together in APG classification. For examples are given here. So if this amendment widens the scope of the taxon, it is shown by sensu amplo or sensu lato, wider if the scope of the taxon is wider. If the circumstances reduces the scope, it is shown by the sensu stricto. You can behind the name you can see sometimes apocynaceae, sensu lacto, apocynaceae, sensu stricto. Example is given here, just to see that. For example, if a genus is divided into 2 or more genera, the original name must be retained with indication that sensu stricto for one of the resultant genera, the one which includes the type species. Again, there is some instance in broader APG circumscriptions of the family Malvaceae, sensu lato includes Malvaceae sensu stricto, and also the families of Bombycaceae, Cerclaceae, and Tiliaceae. I think you are not understanding. So, as per Bentham and Hooker classification, Malvaceae different family, Bombycaceae different family, Cerclaceae different family, Tiliaceae different family. As per Bentham and Hooker classification. Now, the APG classification, what says, as far as molecular evidences, all these families, the species of all these families have share or have the common characters, molecular characters. So they match all the families. Okay, but matched all the families and gave the name Malvaceae. Now there is no Cerclaceae, there is no Bombycaceae, there is no Tiliaceae in APG classification. Similarly, I told you Asclepidaceae, also there is no Asclepidaceae. There is matched with an Apocynaceae. Right? Among these, but the name is maintained, Malvaceae. Okay, but Malvaceae, there is a Malvaceae within this group, earlier name. Right? So, in that case, whichever species belong to the Malvaceae, older Malvaceae, that is maintained as or mentioned as the Malvaceae sensu stricto. That means the Malvaceae's original type specimen is one of the species. And these are other species. And all these are included under Malvaceae, that is sensu lacto. Sensu lacto means there are many families. Sensu stricto means a particular family. Understand? So if you want any doubt, you can ask me during an free time. Next. So rejections, when at what conditions the rejections are made, right? So one is the tautonym, that is tautonym means the specific epithet, species name exactly repeating the genus name. Malus that is not accepted as per the botanical nomenclature, but zoological nomenclature it is accepted. Naja for Cobra, that is accepted, but the botanical nomenclature it is not accepted. And autonym, that is it is automatically created tautonym for intraspecific, right? For example is given here for Pocosumum travangoricum. So earlier Pocosumum travangoricum was given, the name was given by Beddoe, but later the scientists of Botanical Survey of India described a new variety, right? Pocosumum travangoricum daviculomensis. Near Munnar there is a place called Daviculum. So there they found that new species and they give the name, variety name, Daviculomensis. But the original Pocosumum travangoricum, that becomes automatically variety travangoricum. So that is the auto name. So superfluous names are rejected. That is when new names are given for taxa already having legitimate names, and later names are rejected. Okay, if already one name is there, we should not give the same name. Next slide. So synonyms, no, it is a rejected name due to misapplication. So for example, it's given here for Nitpaya Nauchali. So the year of application is there. You can see the year of applications in nomenclature. Tomorrow you are going to work out on nomenclature. So nomenclature, when the nomenclature issues are coming, you have to see first the year of application for rule of priority. Okay. So the three names are given here. Among these three names, which one is accepted name? So based on the rule of priority 1763, right? So Nafia Nauchali is accepted name. So remaining, but these two names are also given by different botmaster. These two names become synonym. Okay. So as per the rule, it is not accepted, but people sometimes some places they are using the names. Okay. And homonym, there is the names exactly it is repeated. Giuseppus, Giuseppa, but different authors, different place, different years. In the later homonym is rejected. Okay, so homonym means exactly the species and the genus name are given to another species. This is a thing. Next slide. Okay, just for one problem. Just very simple. This is among this 5 name. Which one is the correct name? As per the rule. This is the final slide. I think many people are looking at the watch. Which one is valid name as per the rule of nomenclature? Huh? Origanum? Bengalensis. Is it correct? Is it correct? What I told you that when the name is changed to another genus, one genus to another genus, the specific epithet must be retained, the species name must be retained. Right? In nomenclature problems, two things you have to see first. One is year of application and the species name. As per the rule of applications, which one is the earliest published one? Origanum? Bengalensis. Okay. Is there any other Bengalensis species name? Pogosumum bengalensis. And if you see the repetition of the species name, next to the species name, you have to see the author citation. Whether the first proposed person signed in a botanist name is within the bracket. In that case, that is the correct name. That means Origanum bengalensis name is modified. This is changed from one genus to another genus. This is one example. So like that, tomorrow afternoon you are going to see many problems. Okay, so Dr. Narasimhan will teach you the nomenclatural problems. So this is for your basic understanding for the plant nomenclature, and if you have any doubts, you can discuss during the free time. And tomorrow also, anyway, you have the lecture on nomenclature. And understand that this is very, very important in taxonomy. If you understand this, to my, for me, taxonomy is a very, very simple subject. Some sound is coming from like that. So basically, I'm a taxonomist, but I am not working on taxonomy. So listen. So I'm working on bioprospecting. That is, I'm working on aromatic plants. Right? But my foundation is taxonomy. I'm not going to anyone, any taxonomist for identification of plants. Because basically I'm trained. That's why I'm telling you that first learn taxonomy. In future, you can work on any field. Right? So in my experience, my experiments, I never missed the identification of plants. Right? Even after confirmation, I may cross-check with my mentors. So today, Dr. Narasimhan is coming, who is my teacher. So during my PhD time, Coolly taught me all those things. Okay, so you'll have a very nice time tomorrow. So today, let us break for lunch, and in the afternoon, I think there won't be any boring lectures, right? So there will be a training session on botanical tea preparations and the herbarium techniques, how to prepare herbarium specimens. Okay, so thank you for your patient sharing.
Any doubts there?
Dear participants, if you have any doubts, kindly clarify with them.
But this is very hurriedly presented. Actually, for my student, I teach nomenclature for nearly 4 to 5 hours. That's within 1 hour. So I presented here plant identification and nomenclature also. Even 4 to 5 hours also not sufficient to teach a nomenclature, right? So if any other occasion is now available, I will teach you more. Okay, thank you. If there is no questions, anything, then thank you very much. See you in the afternoon. Thank you.
Thank you, sir, for sharing the various types of identification methods and classification in the field of taxonomy. We came to know that how we can identify the plant just by observing their peculiar characters. Your talk on binomial nomenclature and nomenclature type makes it understand simple and easier. Thank you, sir.
Kindly join us in lunch arranged in Fatima Hall and assemble back at Botany Laboratory at 2.